Hi everyone, so my name is Caitlin O'Dell and today I'm going to be talking about cystic fibrosis. So just a little introduction into cystic fibrosis. Patients that are diagnosed with this produce mucus that is abnormally thick, and this interrupts the function of vital organs, one of these organs being the pancreas. And this can lead to decreased nutrient absorption, so a lot of CF patients struggle to gain weight. Negative effects to the airways are the most common cause of death, and you can see to the picture on your right, you can see a clear airway with healthy lungs and then the thick mucus next to it, which is caused by CF. And it's also the most common lethal genetic disease for individuals of European descent, and it's caused by a mutation in the cystic fibrosis transmembrane conductance regulatory gene, and this encodes a chloride channel. So actually a lot of CF patients will have very salty sweat because it's not being regulated by this gene. And a little bit of history about cystic fibrosis. So it can actually be dated back to 1595 when Peter Paw, he was a professor of botany and anatomy at Leiden, he reported an autopsy of a bewitched 11-year-old girl, and he actually reported the exact symptoms of CF, so it can be dated back to that long ago. And Dorothy Anderson was the first person to identify the symptoms of cystic fibrosis in 1938. So it was actually recently identified, it wasn't too long ago, and you can see pictures of them to your left. And in the 70s and 80s, treatment options became available, such as antibiotic treatments, organ transplants, and oral steroids. However, during this time, so the 70s and 80s, identifying the gene was still being researched, so nobody really knew the cause of this. And into the molecular genetics of CF. So the Cftr is a gene that acts as a chloride channel, and it contains 4 domains. They're large peptides that fold independently, so there's 2 symmetrical halves. And the halves contain a membrane spanning domain 1 and 2, and then an NBD 1 and 2. And the domains regulate the gating of Cftr, so essentially the opening and closing of this channel. And when the Cftr gene loses function, it can lead to impaired chloride, bicarbonate, and liquid secretions, and that's the mucus you see. And to your right, you can see what that kind of all looks like. You can see the membrane spanning domains and just the different parts of this gene. And the mode of inheritance for cystic fibrosis, it's an autosomal recessively inherited disease, and you can see on this pedigree I put in, it's a little blurry, but it's a good representation. You can see if 2 carriers carry that gene, there's a 25% chance that their child will have CF. And those carrying the gene do not have cystic fibrosis. And it's autosomal, so that means that the mutation sits on one of the autosomes or pairs of non-sex chromosomes. That's numbers 1 through 22. And Cftr gene is located on the long arm of chromosome 7, and again, there's a 25% chance of inheritance. And now I'm going to get into one of the most common mutations that cause CF. However, there are over 1,000 possible mutations, but it's the deletion of phenol alanine at the 508th amino acid of the Cftr, and you can see that in the picture. So you can see the 508 location, and then if you have cystic fibrosis, you can see it's missing below. And it occurs in the NBD1 domain, and this decreases structural stability of the domain and its activation energy needed to refold. And this results in defective chloride secretion, sodium chloride hyperabsorption failure, and then the failure to clear mucus. And it's responsible for 66% of cystic fibrosis patients, so a lot of them. Now, there are some treatment options if you have CF. The FDA actually approved a drug called Orkambi, and it includes a Cftr and an ivacaftor. And this treatment option only works on the mutation that I just mentioned, so the F508 homozygous mutation. And the homozygous mutation just means it's the same from both parents. They don't have multiple types of mutations. And the Cftr interacts directly with the MSD1 domain, and it partially rescues the functional mutated proteins. And this happens because some of those mutated proteins pass cells quality control, and they actually have partial function. And the Cftr facilitates the opening of the CFTR channel that CF patients can't do on their own. And it's shown to increase pulmonary function and increase lung health and also mucus clearance. So in the picture, you can see Cftr is helping the protein stay open longer, so things can pass through it, and then the lumacaftor brings more of the CFTR proteins into the cell's surface. There's also another treatment that works on a molecular level. This is the Crispr Cas9, and it's a method in which DNA of cystic fibrosis cells is directly corrected in a way that subsequent cells produced by replication carry a working version of the CFTR gene. And it's a form of gene editing where it edits out the mistakes in the CFTR producing functional proteins. So you can see in the picture, they essentially are cutting both sides of the DNA, and then it's being replaced by the correct amino acids. And a great thing about this type of treatment is it works on other mutations, unlike Orkambi. And however, there are some downsides to it. It's believed to only correct half of the lung stem cells, and editing in the wrong place in the genome can cause cancerous mutations. And it's still in testing, so there is a possibility that those problems can be improved upon and they won't be a risk. And this is just my works cited, so thank you so much for listening.